Good afternoon, everyone. Thank you for joining us for what is our last NU at Noon session of this season. It's hard to believe that we're already at the end. So I just want to thank everyone for joining us today. Just a couple of quick reminders. We are recording this presentation just so that you're aware and also so that you can view it in the future. Please remember to keep your microphones muted throughout the duration of the presentation. And we will be doing a Q&A session at the end. And you can submit your questions via the chat box when we open that up. So without further ado, I'm going to turn it over to Joe Hunt to introduce our presenter.
Well, thank you, Ashley. While the first true man-made antibiotic, penicillin, was manufactured in 1928, naturally occurring antibiotics have been in use for thousands of years. For example, around 2500 BC, the Chinese were using moldy soybeans to treat conditions like boils, pimples, and abscesses. The customary alternative to natural molds for treating infections was bloodletting, using knives or leeches. Although today's manufactured antibiotics are potent, they are only effective in treating bacterial infections. This inherent limitation has often led to misuse and abuse by both doctors and patients. As a result, today's bacteria are rapidly responding with new, highly resistant strains, so-called superbugs. Our presenter, Dr. Brandon Dionne, will not only share with us more about the interesting history of antibiotics, but also what is now necessary to ensure their future effectiveness. Dr. Dionne earned his Bachelor of Science in Human Biology from the University of Southern Maine and a doctorate of pharmacy from the University of New England. He completed residency training at the University of New Mexico. Prior to joining Northeastern, Dr. Dionne was an assistant professor at the Midwestern University College of Pharmacy, Glendale. Here at Northeastern, he is currently an assistant clinical professor in the Department of Pharmacy and Health System Sciences. Additionally, he currently practices as a clinical pharmacist in infectious diseases at Brigham and Women's Hospital. So a warm welcome to Dr. Dionne, and to begin the presentation, I turn it over to him.
Well, thank you for that introduction. And so you covered some of the history that I'm going to talk about with antibiotics. I was impressed by the knowledge on the moldy beans. So we're going to review some of the history of antibiotics and how they were first developed, discuss the problem of antimicrobial resistance, and identify some potential strategies to combat antimicrobial resistance and preserve antibiotic use. Bacteria and other microbes like fungi have been at war with each other for centuries. They produce natural antibiotics to compete with one another. So of course, most of our antibiotics are going to come from this source, from compounds that are produced by bacteria or fungi. And so before we could identify and synthesize these, people actually understood that there were possibilities of using the mold, as Joe talked about with moldy beans. And so there are records of from Egypt, China, Greece, and Rome reporting successful use of moldy bread to treat topical infections. And interestingly, tetracyclines have been detected in human skeletons from as early as 350 CE. So even before we were aware of antibiotics, people were using them to try to treat bacterial infections. The first real modern antimicrobial was arsphenamine, also known as Salvarsan, which was developed by Paul Ehrlich in 1909. And this was used primarily to treat syphilis, and it was really the first treatment for syphilis available. And it's an arsenic-based compound, so there are some concerns with toxicity, but we actually still use arsenic-based compounds today, primarily for African sleeping sickness. So this was unique because it was fully synthetically made. This actually didn't come from natural sources. And so it's not technically considered an antibiotic because antibiotics officially are defined as those that come from a natural source or are synthesized from an originally natural source. The next available antibiotic was sulfanilamide, or Prontosil. And this was also a synthetically made, it actually came from a dye. And that first became available in 1935. And that was really the first antimicrobial that was widely used. And it was originally used to treat streptococcal infections. And historically, it was actually used to treat Franklin Delano Roosevelt's son of a bacterial infection. And this is part of the, some of you have used. Well, you may be wondering, you know, I mentioned some other antibiotics, but the first one that comes to mind for most people is penicillin. And Alexander Fleming was credited with the discovery of penicillin in 1928. And I say credited because there actually were reports of use of penicillium, the mold that penicillin comes from, prior to Alexander Fleming's quote unquote discovery in 1928. Actually, Joseph Lister, who was famous for aseptic technique, wrote about using penicillium mold. And this was actually a kind of a happy accident. So Alexander Fleming was a bacteriologist at St. Mary's Hospital in London, and he went on a vacation and had left some plates out. And when he came back, he noticed that there was mold growing on them, and a bacteria called Staphylococcus aureus was killed in the areas around where the mold was growing. So he realized that there was probably some compound being produced by that penicillium mold that could be utilized to treat bacterial infections, to kill bacteria. Unfortunately, he didn't really ever find a way to isolate penicillin very effectively. And so Florey and Chain at the University of Oxford were the first to discover a fairly efficient method to extract and purify penicillin from penicillium. Basically, they were growing the penicillium mold and collecting the fluid from that growth and extracting and purifying the penicillin. And so it was a very time-consuming process, and there wasn't a lot of penicillin. Initially, they just had small amounts that they could use to treat mice, and they found that it was very effective in treating mice that were intentionally infected. They finally were able to create enough penicillin to try treating a human in 1941. And the first patient treated was actually a police officer in the UK. And the story is that he'd gotten a scratch from a rose, and this is common, something that happens with streptococcal infections, and it had become overwhelming. And so they identified this patient as the first to be able to try penicillin in a human. Unfortunately, they had a limited supply, so they actually were collecting his urine and recollecting the penicillin that had been unchanged, that had been eliminated through his urine, to try to retreat him with it. Unfortunately, they didn't have enough, and he died from the infection before the penicillin could cure. Supply was fairly limited of penicillin because of this challenge of actually collecting it, and in 1945, it finally became able to be mass produced. During the war, there was an anecdote that Winston Churchill actually decided to prioritize treating STDs with penicillin because it was a fairly easy thing. It took a limited amount of penicillin and could get soldiers back out in the field faster than any sort of major infection. And one of the challenges with penicillin was that it was actually chemically synthesizing it. And this was first done in 1957 by John Clark Sheehan at MIT here in Boston. And this was not the most important piece of this because the way that he synthesized it was actually not efficient enough to use for mass production. But the important thing was that he developed or identified the core structure of penicillin, which could then be used adding on different side chains and different chemical structure, changing the chemical structure to develop new penicillin-based antibiotics. And so he actually created ampicillin from this, which is an antibiotic, much like penicillin, that we still use to this day. And this is actually a common strategy that's still used is taking an antibiotic with a known activity, changing certain compounds to try to change the activity or the stability. And so this was really the beginning of the golden age of antibiotic discovery. And most of these antibiotics were coming from natural sources. Streptomycin was first isolated from the bacteria Streptomyces griseus in 1944. And so most antibiotics, if you notice that they end in mycin or mycin, they came from Streptomyces originally, or were at least synthesized from a compound that originally came from Streptomyces. Cephalosporins were first isolated, which are another beta-lactam antibiotic, from a fungus called Acremonium, which used to be known as Cephalosporium, hence why they're the cephalosporins, collected from a sewage outlet in Italy in 1945. And trying to get a lot of unique compounds was actually very difficult. So it was expensive to send people out to look for samples. And most of these bacteria and fungi were coming from soil samples from different areas. So Eli Lilly actually had a brilliant idea to ask Christian missionaries to send back soil samples. And one of those soil samples that was sent from Borneo in 1952 actually was it contained the bacteria Streptomyces orientalis, which allowed us to isolate vancomycin. And vancomycin is still one of our broad spectrum antibiotic agents that we use today very commonly. I mean, this is one of the most common antibiotics that I see in clinical practice. In addition to antibiotics, we also identified other compounds that have utility in treating infections. And so clavulanic acid, which is a beta-lactamase inhibitor. So a beta-lactamase is an enzyme produced by some bacteria that can break down our beta-lactam antibiotics. That was identified from Streptomyces clavulagaris in 1976, hence the name clavulanic acid. And this is still used today if you've ever taken Augmentin or Amoxiclav. And so during that heyday, you can see there were a lot of new antibiotics that were developed. So the sulfonamides were really that first class with Prontosil, followed by the beta-lactams, the aminoglycosides, streptomycin, which we talked about. And chloramphenicol was actually the first fully synthetic antibiotic. So it wasn't originally a natural product, but it was synthesized from a related natural product. But what you'll notice as well is that shortly after antibiotics become available on the market, or even sometimes before, in the case of penicillin, we see resistance develop to these antibiotics. And so pretty much it's like whack-a-mole. As soon as you develop a new antibiotic, resistance will develop to that antibiotic. In some cases, it is very relevant. So penicillin resistance developed very quickly in Staph aureus. And so we essentially have no role for penicillin for Staph aureus anymore. But vancomycin, even though resistance is observed 2 years later, it's not very widespread, at least not for the organisms that we commonly use it for. So we do see resistance sometimes that is not clinically relevant. But what you'll notice is that the last unique antibiotic or unique class to be developed was actually daptomycin, and that was in 2003. And so since that time, we've had no unique classes of antibiotics developed. Every additional antibiotic has been some tweak or advantage on a already existing class. The one sort of exception to that is there's a new antibiotic that belongs to the pleuromucilin class that was originally used in livestock that's now been adapted for human use. But this is concerning because without new mechanisms, the bacteria can easily develop resistance. And the challenge with antibiotics is that it doesn't just affect the patient that's being treated, it affects everyone. It's really a tragedy of the commons. And Fleming was fairly prescient on this. And one of his quotes about this was, the thoughtless person playing with penicillin treatment is morally responsible for the death of the man who succumbs to infection with the penicillin resistant organism. And so the question is, what can we really do to prevent resistance? And how does it happen anyway? How do bacteria actually become resistant, or how do infections caused by antibiotic resistant bacteria occur? Well, germs are present pretty much everywhere. Most of them are not resistant to antibiotics. But when you expose someone to antibiotics, a person, or in the case of livestock, or even sometimes we use antifungals in fruit production. So when they're exposed to the antimicrobials, they kill those that are susceptible, leaving only the resistant bacteria. Now, those can grow and become the predominant strain of bacteria, and now you have a resistant infection. Alternatively, some bacteria can actually transmit their resistance to other bacteria, mostly through a mechanism called plasmids. But there's also other types of transfer, like through bacteriophage viruses. And it's important to think about how these mechanisms work, and because how they develop is how we can potentially overcome them. So in some cases, the germs can change or destroy the antibiotics with enzymes that break down the drug. And so I mentioned the beta-lactamases before. And so what we can do in that case is add something that blocks that enzyme, like a beta-lactamase inhibitor. Or we can potentially change the structure of the beta-lactam, like I talked about with penicillin and ampicillin, so that it's more stable to that beta-lactamase or enzyme. Alternatively, bacteria can prevent the antibiotics from even entering the cell to be effective. And again, usually in that case, we have to either give higher doses or change the structure of the antibiotic slightly to make it better able to penetrate the bacterial cell. Bacteria can also use pumps or efflux pumps that can reduce the concentrations of the antibiotic at the site of infection. And so for these, again, we can potentially change the structure of the antibiotic or give higher doses. One of the most concerning changes is if the bacteria actually changes the target of the antibiotic. And so in a specific group of Staph aureus called methicillin resistant Staph aureus, this is actually its resistance mechanism. So it changes the target for beta-lactams so that the whole class essentially is completely resistant. We have developed a new beta-lactam that actually targets that specific change, but you have to develop that. You can't just use the already existing antibiotics. And then the last is that the germs can develop a process that just avoids using the antibiotics target. And so this is like finding a way around the resistance mechanism. Like in the case of Bactrim, it can actually bypass the enzyme that Bactrim inhibits. So there are a lot of different ways, and that changes how we combat the resistance. It's also a concern globally. So that this is this concept of One Health, that the environment, animal, and human health is all linked. And that is not any more clear than in the area of antibiotic resistance, because animal use is actually, based on tonnage, is our highest use of antibiotics in the US. And that's mostly driven by beef and pork. And so when you expose cattle or chickens or pigs to antibiotics, the same process can occur where they select for resistant bacteria. And that may not cause a problem right away in that animal, but their excrement is actually used oftentimes in fertilizer, and that can spread to the crops. Or they can have antibiotic resistant bacteria in their meat products. And humans can be exposed through either of those, or people who are exposed, like farm workers, can also just generally be exposed to the bacteria from animals. And then additionally, if you take antibiotics, you can develop resistance in much of the same way. But there's also environmental exposures. For example, if you're in the hospital setting, or you have family members who are, or you work there like a physician. And so all of these different mechanisms can spread resistance. And so it's a multimodal problem that requires multimodal solutions. I mentioned that the antibiotic heyday was really back in the 1940s to the 1960s, and that's true. So antibiotic development has significantly dropped off, and we'll talk about some of the reasons for that. But this graph shows you antibiotic resistance on the left-hand axis, and this is several different important species of bacteria. But the general idea is that resistance is increasing over time, while at the same time, the number of new antibacterial are being approved by the FDA has been dropping off. You do see that there's a little bit of a bump from 13 to 17, and we have had some new antibiotic approvals since 2017, but most of these are very niche antibiotics, and really don't necessarily answer the problems of some of these resistant bacteria. So we have increasing resistance, but fewer drugs to actually treat these infections. And part of the reason for that is that antibiotics don't make money. So if you start someone on a statin, for example, for cholesterol, they're probably going to be on that for the rest of their life. So they're going to be having to buy that medication all the time. Bacterial infections, though, we treat for a limited duration. So when you have a course, you're only on it for a certain amount of time. In addition to that, most of the antibiotics that we have now work for most people, right? So unless they have a resistant infection, there's not really a good reason to use one of the newer brand only, more expensive antibiotics. And so the use of those is really down from what it used to be. You know, back when some of the cephalosporins were brand only, they were the best options, and when that's what we were using both in the hospital and outpatient. But now we use mostly generic antibiotics, and it costs a lot of money to develop a new drug. And so this graph shows you the profits from antibiotic research. So as they get into their clinical research and finish up their phase 3 trials and get it approved, they finally start to see some profit, but it takes almost 25 years before they actually start to see profit from their drug, and not even be the case with antibiotics. And so those currently in the pipeline in 2015, you can see are mostly low priority drugs, so things that we don't necessarily need. And there's only a few that are targeting some of these higher priority, more resistant pathogens. And part of that has to do with the market. Like they're going to try to target things like skin soft tissue infection that we see a lot more commonly than multi-drug resistant pneumonia. But the problem with that is, again, that we have drugs that already work for skin soft tissue infections most of the time. And this is becoming an increasing problem. So each year, antibiotic resistant bacteria and fungi are expected to cause almost 3 million infections and almost 36,000 deaths in the US. So this is becoming a leading cause of death. And potential adverse effect from antibiotic use is a superinfection caused by a bacteria called Clostridioides difficile, or C. diff. You may have heard about that, but it causes severe diarrhea and sometimes is life-threatening. And so in the hospital setting, it's responsible for 224,000 cases and almost 13,000 deaths. And unfortunately, this used to be limited primarily to the healthcare setting, but increasingly, we're seeing community onset C. diff, and these statistics are hard to actually track. So this data is limited to the hospital, and it probably actually undercounts the true number of cases and deaths from C. diff, which is a consequence of both antibiotic use and antibiotic resistance. And antibiotics are very important for our medical system. So in addition to just treating infections, we use them for a multitude of other purposes. There were over 20 million surgeries performed in 2014, and most of those patients are going to receive some sort of surgical prophylaxis. And if we don't have effective antibiotics, we can't effectively prevent surgical infections, and those could be potentially life-threatening depending on the type of surgery that's occurring. Sepsis, which is an overwhelming bacterial infection. At least 1.7 million people annually present for sepsis treatment, and the primary problem is bacterial infection. And so without antibiotics, we can't treat sepsis. There were about 650,000 people who receive chemotherapy annually in the US, and chemotherapy can cause immunosuppression and increase the risk of infections. And some patients are put prophylactically on antibiotics, and others are treated for infections if they occur. And so some of our cancer treatments would have to be modified if we didn't have antibiotics to prevent or treat those infections. There were over 500,000 patients on dialysis in the US. And patients on dialysis are at higher risk for infection. So we don't use prophylactic antibiotics for this group, but they are more likely to get infections, and we may have to change the number of people on dialysis if antibiotics are no longer active. And then perhaps the most impacted group would be patients who undergo organ transplant. There were over 33,000 organ transplants in 2016, and these patients are on antibiotic prophylaxis at least for the initial period after their transplant, where they're at highest risk with strong immunosuppression. But these patients also, because they're often on lifelong immunosuppression, have much higher rates of infection than the general population. And so organ transplants would essentially be completely impossible if we no longer had antibiotics. So what has the government done to try to combat this? Well, they've known it's been a problem for a while, but they really started to respond to it in 2013. And that's when the CDC released their first Antibiotic Resistance Threat Report. And that kind of kicked things off. It made people more aware of the problem. And in 2014, the White House released the first national strategy to combat antimicrobial resistance. The President's Council on Advising for Science and Technology released a report on combating antibiotic resistance, and the CDC published the core elements of hospital antibiotic stewardship programs. And so antibiotic stewardship or antimicrobial stewardship is the appropriate use of antibiotics. And that's actually part of what I do at Brigham and Women's Hospital. We talk with the physicians to try to limit inappropriate duration of treatment, try to stop antibiotics when there's not a clear infection, and also try to use the narrowest possible antibiotic to help reduce the emergence of resistance. And so initially this was limited to the hospital setting, but the CDC has now actually published core elements for long-term care facilities and for outpatient antibiotic stewardship, because most antibiotics are actually prescribed in the outpatient setting. It's estimated to be more than 80% of antibiotics are prescribed in the outpatient setting. So this is somewhere that's kind of an emerging field still, but something that's being addressed. In 2015, the White House hosted the first forum on antibiotic stewardship and released the 5-year National Action Plan and established the President's Advisory Council on combating antimicrobial resistant bacteria. In 2016, the CDC established the Antimicrobial Resistance Lab Network and launched the Antimicrobial Use and Resistance module. So this allows hospitals to actually report their antimicrobial use and antimicrobial resistance to the CDC so that they can better track this over time, as well as provide the hospital with information about how their use compares to other similar hospitals. And that may give them, help them identify opportunities to reduce inappropriate use. A major step in 2017 was that the FDA released the Veterinary Feed Directive. And so, as I mentioned, animals are actually the highest use of antibiotics in the US. And so this directive advised that antibiotics should only be used under the supervision of a veterinarian to treat active infections or prevent spread of infections, but not for growth promotion, which is how antibiotics were commonly used in livestock. And then there was obviously a change in the president in 2016, and so or 2017. So there was an executive order to continue the advisory committees that had been established. In 2018, the U.S. government launched the Antimicrobial Resistance Challenge, which tried to get partners to help develop novel solutions to antimicrobial resistance, including developing new antibiotics, new diagnostic tests. The CDC published initiatives for addressing antimicrobial resistance in the environment, as we talked about with the One Health initiative. And then CDC released their containment strategy. In 2019, the US actually concluded the AMR challenge with multiple partners. And so we're still seeing some of the effects that came from the AMR challenge, but it is promising. And then they released the second version of the Antibiotic Resistance Threats Report. And just this year, the White House released the National Action Plan 2020 to 2025. So that CDC Threats Report actually lists some bacteria that are considered to be urgent threats, serious threats, and also those to watch. So some of the urgent threats are infections caused by bacteria and fungi that are more difficult to treat. We don't have as many options. So carbapenems, which are a type of beta-lactam antibiotic, have traditionally been like the last line therapy for gram-negative bacteria that are resistant to pretty much all of our other options. And so when you have resistance to carbapenems, you really have no other options up to this point. Now, recently there have been a couple of new beta-lactam, beta-lactamase inhibitors that are active, combinations that are active against carbapenem-resistant Enterobacterales, but things like carbapenem-resistant Acinetobacter, we still don't have very good options to treat this bug. And another concerning thing is drug-resistant Neisseria gonorrhoeae, which is the bacteria responsible for gonorrhea. We're seeing increased rates of STI transmission, especially in the younger population. And as they start to become more exposed to treatment with ceftriaxone, we start to see increased rates of resistance. And we don't really have another good single dose option to make sure that this is effectively treated. So this is becoming an increasing concern as well. And then under the serious threats, the ones that we tend to see more in practice are extended spectrum beta-lactamase producing Enterobacterales. So again, these are ones that are resistant to some of our more commonly used beta-lactams. And often we can use the carbapenems for these, but we have seen increasing rates of this type of bacteria. So that is concerning because as we use more carbapenems, we're going to see more resistance to carbapenems. Vancomycin-resistant enterococci also does occasionally occur, although this tends to be in patients who are already exposed to multiple different antibiotics and chemotherapy. So it is a problem, but we still have some solutions for this. Multi-drug resistant Pseudomonas aeruginosa is another one I see in practice quite frequently, and some of the newer drugs that have been developed in the last couple of years have activity, but this is still a major concern. If you see cases where it's extensively drug resistant and we really have no options. So there are some cases where we have essentially no active drugs. And I didn't get into this, but one evolving area to treat these is actually bacteriophages, which are viruses that infect bacteria. And so there are some centers throughout the US that are developing bacteriophage combinations that can be used to treat completely drug-resistant bacteria, which is pretty cool. And then the last major one that I want to talk about is methicillin-resistant staph Staphylococcus aureus, or MRSA or MRSA. You may have heard of that one. And it's commonly transmitted in the hospital setting, although there is some community spread as well. The hospital strain and the community strain are a little bit different from one another. But this is a challenging infection, and it's associated with higher rates of mortality than methicillin susceptible Staph aureus, which is susceptible to our beta-lactams. But we do still have quite a few options at this point that can treat MRSA. So these more serious threats, we still have some options for. These urgent threats, we have very limited or sometimes no options for. One of the strategies is to potentially create new antibiotics. And I had mentioned that there's very little incentive to do that, right? Because you don't make a lot of money back on these antibiotics. So in 2012, the Generating Antibiotic Incentives Now, or GAIN Act, was passed. And that created this category of qualified infectious disease product. And this is certain antibiotics that treat targeted infections, mostly those that are on that urgent or serious list. And what it does is provides 5 years extra exclusivity on their patent, as well as gives them a fast track designation so that they can get FDA approval more quickly than is typical. So they can help to get it on the market more quickly, and then they have sole rights to it for longer. And that's considered a push incentive. So that incentivizes the drug companies to put their products out onto the market. But the problem is, once they get out there, there's not necessarily a lot of use for these, right? So if you're developing an antibiotic that targets carbapenem resistant Enterobacterales, for example, we don't see a lot of that. So it's very important that we have it, but we're not going to use it very often. And we want to preserve it for those cases that we do need it. So you also need what's called a pull incentive. So something to keep it on the market once it's out there. And the UK has really been leading on this, but in the US, there is an act that has been proposed called the Pasteur Act, or Pioneering Antimicrobial Subscriptions to End Up Surging Resistance. And sometimes I think if they spend as much time passing legislation as coming up with the names for it, we would be in a better place right now. But this is kind of cool. It creates what people are calling a Netflix model for antibiotics. So it's a subscription model where the government will basically guarantee the companies certain revenue on these antibiotics regardless of use. So it delinks use and reimbursement, which is really important because we don't want these to be used very often. So, and it also helps reduce the cost prohibitive nature of using these antibiotics. So in some cases, I work at the Brigham and we have access to these medicines. Like, we can use a more expensive, newer agent to treat an infection because it's a better agent, but some hospitals don't have access to that. And so they still are stuck using older, potentially more toxic antibiotics. And so this may also help to solve that problem, because the hospital would not be liable for the cost of that medication. The other advantage is that this would incentivize hospitals to actually report to that antimicrobial use and resistance module that I mentioned before, so that the CDC can better track our both our antimicrobial use and also our resistance rates. Right now it's voluntary, but this would allow hospitals to bill separately for these expensive drugs if they establish stewardship programs, antimicrobial stewardship programs, and report to this module. So it still hasn't passed yet, but I'm hopeful that this bill or something like it will pass in the near future, because it is very much needed. I think the other thing is we'll have to keep an eye on the UK, who has already actually adopted this subscription-based model, and see how it works there. But I will tell you, we have one example of this working not for antibacterials, but for medications that treat hepatitis C. Louisiana actually struck a deal with the company that makes the leading hep C treatment, where they paid a set fee to treat as many people as they could. So rather than paying per use, they just had a set cost. But it also helps with the company because now they have a guaranteed revenue. So there is a benefit to this for both sides. We can also potentially use novel technologies. So the part of the limitation of developing new antibiotics and why we've really just been nibbling around the edges, like adding new types of beta-lactamase inhibitors or slightly tweaking the structure, is because it's difficult to identify new compounds. We've really hit the limit of things that we could previously grow in the lab. And so a couple of researchers here at Northeastern, Drs. Kim Lewis and Slava Epstein, developed this technology called the iChip. And the cool thing about this is that it can culture bacteria that previously couldn't be grown in a lab. And that allows identification of novel antibiotics. And so a couple of years ago, there was a news story about this antibiotic called Teixobactin, which was actually identified from a soil sample from Maine, which is where I grew up. So it was really cool that it was at Northeastern and a soil sample from Maine. But the specific compound, the Teixobactin, which is active against methicillin-resistant Staph aureus, is not super exciting to me, but this technology and the possibility to identify new novel compounds is very exciting. The other strategy that people use is something called high throughput screening. So now that we have much better computing power than we did even 10 years ago, they can use computer modeling to make tweaks to antibiotic structures to predict which ones might have good activity. But the challenge there is even once you identify compounds that have activity, they can potentially be toxic in vivo, in animals or humans. And so even if you identify a compound with good activity, it might not make it through to actual clinical use. And because of that, newer is not necessarily always better. And there's a quote which is somewhat, we try not to label patients any longer. So instead of calling someone an alcoholic, we would say that they're someone that has alcohol use disorder. But this quote really, I think, sums up what I'm talking about. And it's the development of new antibiotics without having mechanisms to ensure their appropriate use is much like supplying your alcoholic patients with a finer brandy. So even if we develop these new antibiotics, bacteria are going to become resistant to it eventually. And so we need to come up with other strategies. And so stewardship is really that major strategy. And it works. There's hope. Since the CDC implemented the first AR threats report and we started to see more of a focus on antimicrobial stewardship, it's now actually a requirement of the Centers for Medicaid and Medicare services for every hospital to have an antimicrobial stewardship program. We've seen a decrease in vancomycin-resistant enterococcus, in multi-drug-resistant Pseudomonas, in MRSA, carbapenem-resistant Acinetobacter, which, as I mentioned, is one of those major concerning ones because we don't have a lot of options for it. And CRE has remained stable. So carbapenem-resistant Enterobacterales has remained stable. And overall, we've seen 18% fewer deaths from antibiotic resistance and 28% fewer resistant deaths in the hospital. So stewardship is working. Another place that we can target is antimicrobial use in animals. And while we have some responsibility in that as consumers, the largest purchasers of meat are mostly fast food chains. And so the National Resource Defense Council actually surveys and scores the chains based on their antibiotic use policies. They originally started focusing on chicken, and they've now had the majority of restaurants commit to purchasing chicken raised without antibiotics within the next couple of years. So they're no longer even scoring that. And that's very important because reducing antibiotic use in livestock decreases the risk of drug-resistant bacteria developing and spreading to humans. So you will see, some of those that are against this push back and say, well, there's no antibiotics in the chicken when you buy it, right? And they have to test for that actually before they can sell it. But it's not the presence of the antibiotics that we worry about, it's the presence of the drug-resistant bacteria because they were exposed to antibiotics, especially for growth promotion. And so chicken has largely been resolved at this point, or is on its way to being resolved, but beef, pork, and turkey still lag significantly behind. And so this is the National Resource Defense Council's scorecard for chains based on their policies around beef. And so Chipotle and Panera have traditionally always been pretty good about this. McDonald's actually recently improved by committing to purchase beef without antibiotics within the next 5 to 10 years. But you can see most of the chains do not have good policies on this. And we'll finish up with what you can actually do, right? So we talked a lot about what the government's done and what chains can do, but what can you do? Well, one major thing is to only take antibiotics when they're necessary and exactly as they're prescribed. And so this chart shows you some of the common infections that we see and whether or not they are caused by a bacteria, a virus, or potentially either. And as you can see, antibiotics really are only necessary for bacterial infections. They don't treat viruses, as Joe mentioned. So strep throat, whooping cough, and urinary tract infections, they certainly require antibiotics. But things like sinus infection, middle ear infection, which is more common in children, and bronchitis, even if they are caused by bacteria, don't necessarily need antibiotics. And so there's certain cases where we would use those, but it really has to be decided in consultation with a physician. And then things like the common cold and sore throat and flu are always caused by viruses and do not need antibiotics. And the flu, we actually do have an antiviral that could potentially be used. So these do not need antibiotics. Another major thing that you can do is get recommended vaccines. So some of our vaccines, like the pneumococcal vaccine, protect against bacterial infections. And so if you never develop pneumococcal pneumonia, then you won't have to be treated with antibiotics to then potentially select for resistant bacteria. So by preventing infections from occurring, we're preventing exposures to antibiotics and therefore resistance. Using proper hand hygiene. So making sure that you clean your hands. This is helpful for reducing viral infections, but also bacterial, and also preparing food properly. So if you touch raw meat, which can potentially have drug-resistant bacteria, make sure that you're appropriately washing your hands, cooking it to the appropriate temperature. And you can buy meat that's been raised without antibiotics. So you can patronize chains that have committed to this, but you can also buy meat for yourself that has the label of raised without antibiotics ever. And then the last thing you can do is advocate for some of these incentives for new antibiotic development, like the Pasteur Act. And so there are forms online that you can submit to write to your congressperson to actually support the Pasteur Act so that we ensure new antibiotics are continuing to be developed to fight these drug-resistant bacteria. And with that, we'll take any questions.
Great, thank you so much. Yeah, and if anyone has questions, please feel free to drop them in the chat box now and I can read them aloud. So I'll give everyone a few minutes to type. Okay, so our first questions. What about antibiotics for things like Lyme disease or related conditions?
So yeah, Lyme disease is, antibiotics are very effective if it's treated up front, but I'm wondering if they're asking about something like post-treatment Lyme disorder. So patients after they have Lyme can have these chronic symptoms, which are thought to be an autoimmune reaction to proteins on the bacteria. And so there are some cases where people are treated for long periods with antibiotics for this quote unquote chronic Lyme or post-treatment Lyme disorder or Lyme disease syndrome. And they've actually done randomized controlled trials comparing a long course of antibiotics versus not, and there was no benefit to the long course. And actually, a couple patients in the antibiotic arm died from bacterial superinfections. So there are a lot of harms to antibiotics. So if you don't absolutely need them, it's important not to take them. But yes, if you have acute Lyme infection, treating it as soon as possible is very important to help prevent that the transition into post-treatment Lyme disease syndrome.
Great. All right, any other questions? All right. I guess your presentation was so thorough. We don't have any. I just want to thank you so much for presenting today. It was fascinating. You provided so much information to us. So I just want to thank you. This was great. And I also just want to thank all of our attendees as well, whether this was your first session or you attended all of our sessions. We really appreciate it, and looking forward to seeing everyone in the new year for our next round of NU at Noon programming.
Yeah, thank you everyone for attending. And if you do have any questions, my email is on the Northeastern website, so don't hesitate to reach out.